Okay, my afternoon. So, good morning to you who are in other time zones than me. Good afternoon to those who are on the East Coast. I'm Michelle Dunn at the National Institute of Health. I'd like to welcome you to the second semester of the B2K Guide to the Fundamentals of Data Science. Now, last week, and for the next few weeks, we're about computing. So last week's talk was Professor Kovac, who gave an overview of the area. This week we'll have a narrower topic, but still at a overview level, a high level, 10,000-foot view of the specific topic of workflows and pipelines. So we'll hear about what scientific workflows and pipelines are, and how they will be useful or even critical to science and big data management. Our guest lecturer for today is Dr. Rommie Amaro, who is a professor of chemistry and biochemistry at the University of California, San Diego. Her research is in computational methods for drug discovery, and at San Diego, she oversees a lab that does large-scale simulations of increasingly realistic biological systems in order to investigate protein function. She has received many awards, including the NIH New Innovator Award, as well as a very prestigious PCASE Award. PCASE stands for the Presidential Early Career Award for Scientists and Engineers. Dr. Amaro has received many NIH grants. Is the director or co-director of two very large NIH awards. One is for the National Biomedical Computation Resource, and another is for the Drug Design Discovery Resource. In addition, she has received a BD2K R25 award to build an open educational resource, which is a couple of MOOCs, as well as some a way of discovering different materials. So to find out more about that, you can go to Rommie's website. You could also go to the Big Data U website as well. So please join me in welcoming Dr. Rommie Amaro for this lecture today. Rommie?
Thank you, Michelle. So I'm very happy to give this lecture in the BD2K series. I'm going to be introducing a topic that is sort of close to my heart as a biomedical researcher, and that is the topic of workflows and pipelines in biomedical research. So just to recap, you just heard an introduction from Michelle, but I'm a professor of chemistry and biochemistry at the University of California, San Diego. I direct an NIH P41 multi-scale modeling resource called the National Biomedical Computation Resource. I'll tell you a little bit today about some of the activities there. I'm also the co-director of the Drug Design Data Resource, which is another NIH drug design methods development, community-oriented resource. And I think the main thing that I want to get across in this introduction slide is that I am a domain scientist. As you can tell from my background, I do work, obviously, in data science, and there's many very important aspects that impact what we do. I'm also a workflow convert. And so what I want to do in today's seminar is, because my understanding is we have a pretty broad audience in terms of attendees, so I've tried to make this a pretty general talk all about workflows and pipelines, so I'm going to give a broad introduction to workflows and pipelines. I'm going to explain why they're both useful and critical to what many of us are trying to do in the biomedical research enterprise. I'll showcase, by way of a specific example in biomedical research, how workflows can be useful for what we're doing and what we, for enabling reproducible results. And I will also introduce an example workflow tool that we've developed at one of my resources, as well as discuss training opportunities and additional material for more learning beyond just the introductory concepts that I'll speak to today. So I think probably most everyone knows, I'm sure on this call, that it's an extremely exciting time for computing in biomedical research, and that's really for a number of reasons. One is, of course, that we've had a tremendous increase in computing power. And so what I'm showing here is just the increase in compute power sort of as a function of time, and that's been available on the national supercomputing resources. So the supercomputing resources are these large architectures that are available for academic researchers in the United States to perform various types of high-performance computing applications. And biomedical research has certainly benefited significantly from these resources. So this is just showing how in the early 90s these first computing resources came online. They had about 12 CPUs, and about every 4 to 5 years since then we've increased our computing power by an order of magnitude, taking us to 2017 now actually, where we're really right now in what we call the sustained petaflop performance capabilities, and that's brought to us by machines like Titan at Oak Ridge, also Blue Waters at NCSA. And something very critically important has been actually the rise in graphical processing unit technologies, or GPUs, which have really brought supercomputing to the desktop. And so a lot of the algorithms that we use in biomedical computing research actually port very well to the same architectures used to develop great graphics for video games. And so this has really also been a game changer in a number of ways for biomedical computing. And then, of course, I think I'm sure others in this series have mentioned that there was recently, they've initiated the National Strategic Computing Initiative, or the NSCI, in order to bring exascale computing into reality, hopefully maybe within the next 5 years or so. So just a tremendous increase in computing, and that's really infiltrated many, many different aspects. Of course, not only in biomedical research, but for the scientific enterprise, generally speaking. And of course, it's not just computing, but it's also data. Access to a number of different types of data, everything from proteomics and genomics to increasing data availability for structural biology at numerous resolutions, including things like from the structural data for molecular componentry, like in the PDB, as well as macromolecular complexes, thanks to advances in direct detectors with cryo-electron microscopy and so forth. And so in terms of what we can do in biomedical research, trying to bring together all these different data streams to create new models, for example, either structural models that might integrate proteomics, genomics, high-resolution structural biology details, as well as cellular substructure determined with electron microscopy to bring together to make a multimodal, multi-scale model. There's many, many examples of how data is infiltrating the research enterprise. And so together, both computing and data are really transforming how we do research. And so when we think about how things used to be, I mean, we're not just necessarily doing one experiment at the bench, right? I mean, when we're posing a problem or a question in biomedical research, it's much more than just one step. A typical or a state-of-the-art example would be starting with electron microscopy and all the terrific advances there in terms of capabilities and resolution, for example, with macromolecular complexes. So we start with our data, we collect the data at the scope, we then send that data, for example, at UC San Diego on a high-speed network over to a supercomputer. And I'm just using this as an example here at San Diego, where we can take the data directly from the microscopes to the COMET supercomputer in order to do image processing. And then once we have that data, it's ported again through a high-speed network from the supercomputer into the lab. And in the lab, we can build and interact with these data sets using a variety of software resources and develop models and interrogate them. We might add additional components to those models, for example, from a number of different community data resources, just there's many different resources to possibly pull from as you, again, build and interrogate your model. And then we might send that new sort of representation of the data or the method, we might send that back again to the supercomputers, for example, to Blue Waters, and then we have more data that we process and return back and can interact with in various ways. So this is showing sort of a possibility of looking at structures with virtual reality or other sort of more than just in 3 dimensions. And so really where we're at now is where experiment, simulation, networking, and expertise are all combined into a super facility, but it's really sort of transforming how we can do biomedical research. And so the challenge, or one major challenge, when you think about sort of the complicated end-to-end scientific discovery process, a major challenge is the growing complexity of the computation and data that are required to solve these highly specialized, significant problems in biomedical research. And this complex scientific enterprise really brings a number of challenges. But one solution to the growing complexity is the creation of pipelines and workflows that are able to assist in the assembly of data sets, as well as integrate what we call domain-specific software codes that are able to operate or execute on various hardware systems or computing architectures, and to do so in a way that is robust, usable, and reproducible. And this is really what's going to be demanded for, or what is currently demanded and will continue to be demanded for successful biomedical research projects. Okay, so what is a scientific workflow? So scientific workflows emerged as an answer to the need to combine multiple data and computing components in an automated fashion. So a scientific workflow is really basically just a series of different computational steps that scientists use to generate results, okay? And this may involve accessing multiple different applications, scientific applications in different databases, as I mentioned. Also, it could involve the processing of data, most frequently it does, using computationally intensive jobs on various types of computing platforms, and I'll tell you more about those with some specific examples. So the idea is just that it's capturing, these scientific workflows are really capturing, again, sort of the complicated, many step end-to-end experiment, ideally, that is that represents a scientific result. Okay, and so even backing up, sort of getting more simple about it. So really, what is a workflow? And so there's workflows everywhere, of course, not just in biomedical research. So if we just think about when we're trying to cook something. So I think one analogy that I sort of like when I'm trying to introduce the concept of workflows and pipelines, to be very introductory about it, is to think about how one cooks, or how one basically, cooks. So when you're trying to create a recipe and cook food, you have to basically, there's many steps that you're doing. You're shopping, you're preparing the food, you're cooking it, and you're enjoying it, eating it, and you're also storing it. And so each of these steps has, it takes a certain amount of time, and it's essentially a workflow. Some of those tasks that have to be performed might be enjoyable, as sort of is illustrated here with the sort of the beige text. Some of them might be more tedious or chore-like. And so one example is, like, we're making pasta this evening. So there's all these different things that we have to do. It's starting with the shopping for ingredients. You can see, like, the raw ingredients on the left. You then have to prepare the different ingredients, maybe by chopping and dicing, etc. You then have to cook it, and there might be multiple things that you need to cook, and then ultimately you can serve it, eat it, and then store it. So these are all different steps that sort of represent not a biomedical research workflow. I'll get to that, but just sort of the overall concept. And so each of these steps, they take place in a certain order, and they have a certain time requirement. And some of these things need to happen at the same time as others. Some of them needs to be staged. Some things can happen in parallel. Others need to happen in series. So, this is an example that we're all familiar with. So we can think about sort of workflows in this context, even for biomedical research. And so one thing that we might want to, one way that workflows can be applicable here is if you want to speed things up. So if you're cooking, for example, there are things, time management can become very important if you want to actually come up with a result or a cooked product at a particular time, right? So time management is important, and you can take advantage of this downtime when you're working on one element of the recipe. You can then shift your attention to another while something is, while, for example, the pasta is boiling. And so these recipes can be modified and they can be optimized. And also, similarly, analogously, what if you have more than one cook? So can particular jobs be farmed out to different people, or in this case of automated workflows, it's to different hardware architectures, and what can be done at different times or the same times with multiple participants. And so one example, again, going back to cooking, so if we, when we're just starting this, let's say that we have our vegetables, and this is something, so we can sort these vegetables, and if we have multiple cooks, each cook can basically be chopping and doing the work on a certain set of the vegetables. And then now this would be equivalent to a map function where the input are the veggies, and the chopping is what we would call a user-defined function, and the output of this operation would be chopped groups of each kind of veggie. So this is sort of the food equivalent to the map function. Now we have these outputs of these different piles of veggies. And we can think about, now we want to bring them together. So we have all these different piles, we want to bring them together. We bring the tomatoes together, the peppers together, the onions together. This is like similar to what we would call a reduce step. So here now the input is having chopped batches of vegetables for each type, and the equivalent user-defined function would be to combine these vegetables basically based on type, so we can sort them. And then the output is a bowl of vegetables per veggie kind. So we can, again, these are sort of the food analogies of developing functions and how we can sort of parse out different jobs to different people in order to expedite or optimize what we're doing. And then you think about when you want to make something more elaborate. Now we're not just doing pasta. We want to make a Thanksgiving dinner. We have many, many dishes to make. We may have many helping participants. Each, recipe and so forth has different preparation time, cooking time, cooling time, etc. And so as a chef or as somebody who's organizing this, you have to think about all these things. When do you start cooking? What order do you cook, etc., etc. And so this is an example of how we already use workflows and are really familiar with them in sort of our in our just daily life. And now I'm going to give you some more concrete examples of how these workflows can be useful, how workflows can also be useful in science. And so so workflows can take a number of different forms. They can be, for example, very simple pipelines, where one example is that you have data on a local disk, so you choose, you can read and choose the file. You can then create a function that basically does a multiple sequence alignment for you, and then it outputs that alignment into something that does a phylogenetic analysis. And then it can output that it can transfer the output of the phylogenetic analysis into something that where you can actually visualize it with, for example, using trees and so forth. Workflows can also integrate components to analyze local data. So this is an example of a workflow drawn in Kepler, which I'll tell you more about, for computational flow cytometry data analysis. So here, it starts to get a little more complicated, but basically different components and different data components can actually come together to participate in the analysis and of data that's held locally. We can also use pipelines and workflows to integrate many different online tools and several different kinds of databases using complex programming structures. So here's an example of a workflow that's doing some microarray analysis to identify new candidate target genes. And here we start with microarray data. We do a clustering analysis. We can identify the genes. We can then get the sequence retrieval. We send that back out to the NCBI blast search. It brings back other genomic sequences. And then we can look for things like promoters and promoter model generators in order to generate a consensus sequence, which we put back onto the blast search, and then, again, to finally identify new candidate target genes. So it's a way, this is a more sort of elaborate example of how workflows can be used to integrate a number of different online tools as well as online databases in order to help facilitate biomedical research. Workflows can also have concurrent analysis and data management steps that actually are distributed to different computing platforms. And so this is an example that I will speak much more about when I give a concrete example. But here we can actually, what this workflow is doing is actually one of the sort of what we call actors or modules in this workflow is actually being used to distribute or to sort of spawn out many different jobs in parallel on an HPC platform. And then the analysis on each of those can come back asynchronously or synchronously in order to determine the results of the jobs. And of course, workflows can also integrate cloud resources. So they can tie into and connect to not just local data, but data that can be in the cloud or other places. So overall, biomedical research workflows allow us to assemble complex processes easily, transparently access different computing resources, which is increasingly important as computing enters more and more elements of the biomedical research sort of enterprise. It allows us to, in an automated and more rigorous fashion, incorporate multiple software tools. And I'll talk more about also why that's so important again in the example I'll show you. They also help enable reproducibility, which of course we all know is a major challenge, I mean, for science in general, but also for biomedical research, how we can ensure that when we have one student or multiple students working on a project, that then when those students leave and new ones come in, that the protocols are being followed correctly, and that we really knew how we generated the model, but in a very specific enough way that it's really truly reproducible. And of course, the best research workflows will really build around a community development model, and this is something that I think many fields are now, many subfields within biomedical research are really embracing, and that's the idea that so I can develop workflows in my lab and share them very easily with colleagues and with the community, and we can use each other's efforts to build, again, and use these sort of more elaborate, reproducible frameworks for science. And so there are many scientific workflow systems, as one would imagine. This is something that people have been working on for at least 2 decades. And so one of the earliest scientific workflow systems was something called DiscoveryNet. And then there were a whole bunch like Triana, Pegasus, Kepler, Taverna, Pipeline Pilot coming on in the early 2000s. KNIME is another very popular workflow system. We have BizTrails, Gene Patterns. So you can see sort of the evolution of these through about 2010. And what I'm going to mostly talk about today in the concrete examples is a workflow system called Kepler. This is, again, a very popular common workflow framework. It has a diverse library of scientific components and use cases. It allows transparent support for multiple workflow engines, so you can actually connect. So even within Kepler workflow, you can actually call other workflow systems, for example, like KNIME, in order to execute an end-to-end process. And it's used by many communities, not just in biomedical research, but also in wildfire detection and all sorts of different things. So it's been a useful community development tool. So that's the one that I'll mostly focus on today. But I just also mention, of course, that there are many different programs, and a lot of them have strengths and weaknesses, and you know, there's I'm sure many other learning opportunities if you want to dig in on the details of these other workflow systems. Right, so how do I use workflows, and how did I come to sort of seeing the light with how useful they can be and how important they actually are to the scientific enterprise? So this example I'm going to tell you about is one that's in my domain of expertise, which is computer-aided drug discovery. So the work that I do in my lab and that we do at some of my resources actually involves the computational modeling of atomic level systems in biomedicine. And so we like to think of, one of the major approaches we use is something called molecular dynamics simulations. And I'm showing here a picture of a computational microscope. So this was a sort of a phrase coined by Klaus Schulten, which basically is sort of, we're trying to use these computers, the HPC computers mainly, but now actually it's evolved into using also GPU architectures. We're trying to use these methods to gain additional insight into these molecular systems that we sometimes cannot access with experiments alone. And so what these simulations involve, so basically we're representing all the atoms of our system. We're representing our system at the atomic level, and so we define what we call a potential function, which you can see there. So every 2 atoms that are connected form a bond, every 3 atoms that are connected form an angle, every 4 atoms are dihedrals and torsions, and each of these terms, each of these elements of the system actually has a term in a potential function. And then we're basically using physics, we're just simply integrating Newton's equation of motion over time in order to build up a dynamical trajectory of the time-dependent dynamics of our system. So we're basically representing these molecular level systems numerically and trying to determine the wiggling and jiggling of the atoms, if you will. And so these experiments use a number of different community-developed tools, like NAMD, AMBER, many different tools, actually. And the engine that this microscope runs on are basically the supercomputing platforms and now graphical processing units, and also sometimes local desktops and so forth, but they can run in many places. So one of the major goals was that we had in my lab was to try to use simulation in early stage drug discovery. And what I'm showing on the left is an old napkin sketch from actually when I was a postdoc. And we were just sort of walking through how we could use the output of the simulations, which, as I mentioned, is just sort of the wiggling and jiggling or the time-dependent dynamics of these molecules, how we could use this in computer-aided drug design. And so it's really, you can see this sort of like chicken scratch. And over the course of some years, we were able to actually develop methods that would be gainfully used to incorporate simulation in drug discovery. And so what that would mean is so you can see that I'm sort of advancing the concept here of the workflow is advancing. And this is sort of I think a normal way this is a very sort of natural, normal way for scientific methods and projects to evolve. First, you start with an idea that's literally a sketch on the back of a napkin, and you work on it, you keep working on it, and then you get the first prototype, and then you can continue to elaborate. And ultimately, what I'll show you many slides down the road is that we've gone from basically the napkins type drawing to really a hardened workflow product. And so why do we care? So just in case there are some scientists out there, I want to give you a little bit of science in addition to the computing. So one successful example of this is it involves P53, which is the guardian of the genome. So P53 is a protein that's a transcription factor that's it's mutated in over 50% of all human cancers, and so it's a molecule that senses basically when the cell has some type of damage, like DNA breaks or UV irradiation. P53 should sense that damage and then upregulate pathways related to programmed cell death, like growth arrest and apoptosis and prevention of angiogenesis. And so what happens in over 50% of human cancers is that P53 is mutated and it's inactive, and so these tumors just proliferate. And so we were very interested in trying to understand what was going on there, how the destabilization worked, etc. And a long-standing dream of cancer biologists actually was to try to basically reactivate the cancerous or inactive P53 with small molecules. And so the idea would be that you would have a cancer mutant that has this inactive P53, you would bind some drug to it and you would reactivate it. And then those cells that formerly were just proliferating and metastasizing, that they would actually, re-engage their programmed cell death and die. And so there were a number of compounds that had been found over the years. After about, I don't know, 20 years of research, they had found a handful of compounds. They didn't know exactly how they worked. And so we ran molecular dynamics simulations of this target in order to try to figure out, again, we were interested in looking at the dynamics because we had known that actually it was quite a flexible protein. So this is the output of a molecular dynamics simulation. You can see sort of all the atoms wiggling and jiggling. And what I want to bring your attention to is this area in the middle where we have here, let me see, I don't know if you can see my cursor. But there's a cysteine residue here in a pocket where you see a lot of mobility. Okay, and so what was really interesting from a scientific perspective was that if you look at the PDB database, there's over 100 different structures of P53, and they all have the same sort of architecture around that particular cysteine, which is actually, it's sort of like tucked away and back, sort of a closed pocket. And so what we see in the computational simulations is actually the formation of an open pocket around this residue. And this was very intriguing because we found this new site and we also saw that it might perhaps actually be druggable. So we performed small molecule docking into that site and basically using sort of the work, so here's sort of just a sketch of the workflow for virtual screening where we can take these protein or receptor structures, and this could be coming from the PDB database with X-ray crystallography. It could also be simulation data from the molecular dynamics simulations. We have another database of ligands. In this example, we're using the NCI diversity set. We bring them together using AutoDoc-Vina, so this is another procedural operation. We then do what's called post-processing, and then we pass our predictions to our experimental collaborators for testing. With this workflow, we were able to find one reactivation molecule called stictic acid, and our collaborators showed that there was dose-dependent rescue of mammalian cancer cells. But one of the really interesting things also was that in addition to finding a new compound, we were able to rationalize the mechanism of action for one of the formerly discovered compounds that's actually in clinical trials. So this is really exciting. Then we wanted to scale it up. And by scaling up, we really wanted to be able to screen against many, many more compounds and also to do sort of much more sophisticated post-processing with regard to ranking of compounds. And one way this was enabled was through workflows. And so with this sort of more automated pipeline, so this I'll just call a pipeline because it's really just showing sort of like the procedural steps of what we're doing. We were able to discover many more compounds in a very short amount of time, basically in 6 months, than in all the research efforts of the previous 20 years combined. So it was, once we were able to scale it and with a good model, the discovery really sort of took off. And so what we were able to do was really go from what was just a chicken scratch pipe dream to sort of a pipeline to now a scalable automated workflow. And so, so what does that mean? So when we were doing these experiments that I was telling you about here, this is really, as many people in research will do, and in computational biomedical research, I'm sure, but in many other fields also. Again, when you're developing a prototype, very often what happens is you have a postdoc or a student who has a set of scripts, and they're running these scripts, and sometimes these scripts can be quite elaborate, but there's many, many steps involved in this workflow. everything from selection of the structures that you're inputting into MD to the creation of the molecular dynamics model, running of the trajectories, selecting the structures, how you process the ligands, how you're doing the docking, all of the, there's so many things that one really needs to keep track of. And we do our best. I think everybody, all biomedical researchers try to do their best in terms of keeping very careful lab notebooks, digital notebooks, saving the scripts, and so on and so forth. And that gets us pretty far, but it still has significant limitations, and I'll talk about some of those limitations and how workflows can help us get around those. So what we did was actually to develop using the Kepler workflow system, we were able to develop a scalable automated workflow. So we would basically have, we would be able to, in this one example, we would have all those small molecules that we wanted to screen against. We put those in a directory, and this Kepler workflow would see this directory. It would go into the contents and then actually perform a series of steps that would, what we call, parameterize the ligands, which is getting them ready for molecular dynamics. We can then simulate them and then do processing. And each one of these, so this little box here, this ligand parameterization, is actually represented by, if you actually dig into this one task, it's actually many subtasks, right? And this is really how it is for many different steps in biomedical research. So when we take, instead of using scripts for this antechamber step and this Gaussian step and so forth, we can actually encode it into a workflow such that all we have to do is have a directory of ligands, and then everything else just flows. As the output finishes, it flows to the next, it flows to the next, and there's everything about it is tracked. And I'll tell you more about that. So all the software versions, all of like when you actually ran the jobs, all the library dependencies, it can all be tracked through something called Provenance. And so when we recapitulate these very complex end-to-end experiments into these automated workflows, it brings tremendous benefit, including increased reuse, because it's much easier, generally speaking, for a new student or postdoc to pick up and run these automated workflows if they don't have to stitch together a bunch of sort of loose scripts. It increases reproducibility because we've now really tracked everything and all the dependencies with Provenance. It helps us scale execution, both of the problem and the solution. So I'll talk a little bit more about scaling of execution. So that just essentially means that we can more easily connect to different computing resources in order to carry out the job. And it makes it easy to compare methods. So if you just want to really change only one thing, you can just go in and change one parameter inside one box and rerun it and compare. And it also makes it significantly easier to train students, because now they see, when they sit down for the first time, they can see the whole pipeline and dig in on each module in a way that is simplified. And so one of the steps that we carried out in the very complicated end-to-end process of small molecule discovery, one of the processes, as I mentioned, was just running molecular dynamics simulations. And so we've worked to develop again, a workflow tool using Kepler that just carries out AMBER, what we call AMBER molecular dynamics. So AMBER is an MD engine that's available broadly to the community, and we've developed now a workflow tool that just allows people to carry out research quality molecular dynamics simulations using AMBER GPU processing technology and the Kepler workflow system. And so these are, again, a sort of a very high-level representation of different steps that we have to do in order to carry out molecular dynamics. And so if we look in this one step of something we call minimization, if you actually zoom into this minimization box, there's many steps. It's not just one step for minimization. There's actually 5 different minimization steps. And so what this tool does, again, is in an automated fashion sort of feed from, as the first minimization step finishes, then the output feeds into the second step, and so on and so forth. And then the outputs from immunization go to a stage we call heating, and then to equilibration. And again, in equilibration, you can see it's multi-staged. And then when we get to production, one interesting thing here is that you see that there's all these different little outputs. So it's going, basically when we go to production, instead of just running on one GPU, we actually scale it out to multiple GPUs in parallel. And so this this workflow tool basically allows researchers to do that very, very easily. Okay, and then so it also has sort of a graphical user interface for users. And so again, sort of the whole the molecular dynamics workflow, you can actually, users can go in to each of these little boxes, and at the base level, they can go in and edit the key parameters. Different parameters can be exposed or hidden depending on, because some parameters, as people know in various programs, you basically maybe never change. Others you have to change depending on your system. So this can be configured. So we configured it for molecular dynamics using sort of best practices for the community, but this can be, different levels of detail can be hidden or revealed depending on the application. And then we can, in terms of where the simulations actually run, they have different options. You can have local execution options. You can run on a cluster, on a local cluster, or you can also port out to, for example, the exceed computing resources like Gordon or Comet. And so, so this sort of, so the gray circle here is just sort of showing all the different types of heterogeneous computing resources that these workflows can connect to, and that includes, for example, operating in the Amazon cloud. And even within one workflow, you can have many different computational architectures engaged. And that's one of the advantages of Kepler, is that if I have one step that needs to run on my local GPU, it can just do that, but it can also route to NSF resources, or again, the cloud, or any sort of computing architecture. So we can have multiple architectures engaged within one single workflow. Okay, so so we're getting to the sort of 40 minutes in. So how does one even start conceptualizing a computational workflow? Well, in general, sort of, we would advise to start with the workflow as a black box. And so just treat the whole workflow as a black box. And you have to think of what is the use case and application. So what is the real science question that this workflow is solving, right? So you're going to have here some input data, and then you have some process, and this, is just shown as one giant black box, but it's sometimes many, many steps, and then you have to think about what your output is going to be. And then you have to conceptualize, then you really need to sort of dig into, well, what are my steps, right? So in terms of cooking, this is things like, if I'm working on the pie, I have to, put the ingredients together, cook it, chill it, etc. Bake turkey, make the side dishes. So this would be for Thanksgiving dinner, all the things you need to do. For a scientific application, this might be things like you got to collect the data, the FASTA data, you have to, do the sequence alignment, etc. And so you basically treat each step like a workflow, like a mini workflow. So you want to basically try to reach what we would call, I guess, an atomic functional step, right? So, so you think about all these things. You have to find the data, access the data, maybe you have to move the data, and then once you have the data, it has to be cleaned, possibly integrated, maybe you need to divide it up or pre-process it. And then you get into actually running the jobs. If you have, like, for example, simulations, you are generating data, processing data, and analyzing data. And then you get into data interpretation and so forth, where you're going to interpret the results coming back, do visualization, and so forth. And so this, going back to our cooking analogy, is sort of, we have the shopping stage, the preparing stage, the cooking stage, and the storing stage. And so questions that one wants to ask is, definitely, where and how do you get the data? What format is the data in? How can you actually integrate or subset data sets? And then, of course, how you analyze the data. What is the analysis function? What are the different parameters that are needed to customize each step, etc. And I will say, that when you have an end-to-end, when you have a complex workflow or experiment, it is, like I said, it's challenging to do this, and it requires a lot of time and a lot of thought. But ultimately, these are the kind of things that will enable truly sort of reproducible science. And I think one of the great things about workflows is that once you do this work of really analyzing and this detail and putting things together and standardizing the process, the data and the process, you then have a tool, and that also becomes that sort of conceptualization of the data itself is actually very valuable. In terms of the step for searching for the data, of course, what you need to sort of know what you're looking for at first. So different data formats, and how, sort of the meaning of the data, how it actually scientifically and conceptually relates to the analysis that you're doing. And then it helps if you, of course, for you to know exactly where you need to find that data, so where to find what you're looking for. And here I'm sure there will be other talks in the series where some of these data sources are discussed in more detail, but there's many searchable data archives and repositories, and people are working very hard to make sure these are standardized with the correct data ontologies and metadata standards and so forth. And they can, anyway, the data could also, for example, be coming from your own sensor networks or other scientific instruments, even just in your own lab. Another important component that needs to be considered in general in the scientific enterprise, but also with workflows, is do you have to move data? Sometimes you do need to move data, and then depending on how big it is, that can be a problem. And maybe you need to come up with different ways to access the data so that you don't have to move it. But in any case, this is just to note that moving data can sometimes, of course, be a big challenge, especially for very large data sets, and it's something to think about and to try to avoid, I would say, at all costs if you can help it. So there's various different data access technologies also that can be used if data moving is needed. Again, I guess I sort of already covered this, but we have the ability. One of the great things about developing these workflows is that once you have them developed, it's, at least in Kepler, it's actually not that difficult to change where you are executing the jobs, either the data generation jobs or the analysis or processing. And so you can move around to different computing resources depending on the requirements of your job. And so for example, some steps may be more memory intensive, some may be more compute intensive, some may port very well to GPUs. You can be flexible about that, and you should be, so that you can really gainfully take advantage of as much, so you can get as much advantage as you can of the different resources. And then, as I mentioned, once you do the work of building the workflow, you build it once, and it is a lot of work to build it, but then you can run it many times, and you have terrific possibilities for reuse. So hopefully, the same workflow will support your computational experimental work and dynamic scalability on many different platforms. And the scalability can be based on a number of different sort of data features like data volume and velocity. And this could change depending on those aspects of it. Maybe you need to change something about the modeling based on different optimization criteria. And then also, of course, these workflows, once you build them, you can run it many times, but you can also alter it based on changes in network storage and computing availability. And so these are tweaks that can be made. And then one of my favorite parts, really, of the workflows, I have to say, is the saving and sharing of the reports and final products with your team and collaborators and with the broader community. Right? So computational data science is right in the middle, sort of bridging the gap between the data that is generated and really the key scientific insight. And so bringing everything together in a workflow, in a scalable, automated workflow, like I showed you with Kepler, is something that can be tremendously powerful. And so, I think another sort of aspect of this is that people they should use whatever favorite tools or programs you need to answer the question to implement the methods in your workflow. I mean, there's no reason to feel that that you need to be limited. I think really the possibilities are almost endless in terms of different possibilities to enumerate within your own scientific domain. And then one of the great thing is that the process itself is shareable. So you have, when you build these workflows, they themselves are their own sort of data structure, and you can exchange them. Communities, of course, are working very hard to standardize them. Again, as I mentioned multiple times, they're scalable, and it helps, these workflows also help enable really sort of accountable science, right? So again, sort of not to beat a dead horse, I guess is what they would say, but these scientific experiments involve many different kinds of data coming from different sources. And there's different versions, and this is the other thing that has driven us crazy, is how you can run a particular program, for example, a docking program one day on one particular architecture, and then there's some update that's made, and now it's version 5.7 instead of 4.2, and maybe it didn't break any of the scripts that you used as input, but maybe it changed the results because of something. And it's possible sometimes that trainees and researchers don't necessarily know the fine detail of what's going on, especially for people who are domain scientists and who are pulling together, 20 different tools in one biomedical experiment. And so one of the great things about these workflows is that a lot of that versioning and dependencies and everything, it can be recorded through something called provenance, and tracked, and then and reported and stored, so that people really know exactly what was happening at what given time, and you can follow that data flow. And of course, so that's just on the data and the execution side, but you can really sort of very clearly see and document what processes ran in which order, which were which steps actually depended on the output from the previous step, which could just run in parallel separately, which libraries were used, and also who generated what. So if you have collaborators, it's a very good way to sort of share everything. And so provenance is something that I know has been talked about already, more technically for sure, in the BD2K series. But basically, for those who haven't heard it, provenance basically is something that helps tremendously with what we call research accountability and reproducibility. It's basically, according to Wikipedia, they would define it as the chronology of ownership, custody, or location of a historical object. So when we think about provenance in terms of workflows, this is really sort of like a digital notebook, but on steroids. It contains it actually can contain different information depending on what you set it, so it can be configured to retain particular aspects of the experiment that you think are important, but it can capture everything: inputs, outputs, intermediate results, all the parameters and details of the different components of the workflows, so all those different actors or boxes which I showed you, all those steps, how the steps are linked together, all the different parameters. This can actually be recorded with data provenance, or with these provenance elements. And this is useful because it keeps the association, it retains the association of results of a particular workflow to processes. It also makes it easier to validate and regenerate results and processes because you know exactly how you got them in the first place. It's much easier to compare different workflow versions, do parameter optimizations, etc. And sometimes some of these programs actually allow you to do sort of intelligent or facilitated reruns and failure recovery. So different questions you can ask with provenance, or different questions that are answered with provenance. For example, you can find out how long did this particular workflow run take? What parameter values were used? What different components or processes or actors does the workflow have? How long does one particular actor take? So how long does one step? Where's my bottleneck? And what workflows did a specific user run on a given date? These are all questions that one can dig in. And so, again, there's many reasons why you might want to share workflows. For example, if you're doing collaborative research, if you want to just inform people of progress or formalize application development, etc. And also for publications. I mean, I think one of the also movements that we're trying to catalyze, at least in the molecular dynamics community, is to, which is challenged with, suffers like many areas with reproducibility. We can actually publish, along with our scientific paper, we can publish these workflow protocols, and hopefully, we're trying to develop a community of researchers who are developing these, and so we can all be ensured that we are really performing research in a transparent and reproducible way for the maximal gain for the community. And right, so when you're sharing workflow results, there's different ways that you can share workflows that have with versioning, for example. You can use GitHub to do that. There's also cloud file hosting services, so you can share workflows using Dropbox, Google Drive, etc. And then the other thing that we also do, and I think a lot of others have taken to sort of recently, is to create IPython notebooks that share execution history, input parameters, and results. There's many different ways that workflows can come into the enterprise, and certainly sharing of the results is super important. Okay, so I only have 5 minutes, and I want to at least leave a couple minutes for questions in case there are any. So, but before I leave, I wanted to say this. I know this was a very, very high-level introduction to workflows and pipelines for biomedical researchers. So if you want to get more information, of course, Google is your friend, but I would just give you a couple of leads. So one place where you can get some hands-on experience, you can go to biobigdata.ucsd.edu. There's a scalable, so there is a Kepler class or course there it should be very easy for people to run. It comes with a virtual machine that you can have everything, all the data and everything you need installed, and lectures. You can actually go to this site and retrieve that information and actually go through the course. And if you want to see, this would be, the first course is just on the Kepler software framework. If you want to see an actual application of how workflows can come into biomedical research, we also have a class that we call Data to Structural Models that walks through image analysis and segmentation for electron tomography. And it applies a Kepler workflow for image segmentation and analysis. And so you can get another sort of a more domain-specific example there. And of course, also, I need to give a shout out to Erudite and the BD2K Training Coordinating Center. They also, I just very quickly searched for workflows in Erudite, and you can see that there's 22 different results and a number of different people who have will show and talk about examples that highlight workflows in biomedical research. Okay, and I do have an acknowledgement slide. Of course, I just want to thank the BD2K efforts, the NIH generally, my groups. Ilkay Altintas actually is my collaborator, co-PI on the BD2K R25 award for the Biomedical Big Data Training Collaborative, and she is she's a major developer of Kepler and has been a terrific resource for information on workflows and pipelines. Okay, so I think I have like 2 minutes. I would love to take questions if there's any questions.
Thank you. That was a fabulous talk. There are a few questions.
Great.
And, I'll combine a couple of them.
Okay.
This one is about sharing of data, and you mentioned a couple of different options, FTP and SFTP. Are there any others? Does Internet 2 improve the transfer rates? Is there anything you can, any advice you can give about that?
Speak to, yeah. So I will say that in general, we try to avoid moving data, but of course we do have to move some, and that's a very good question. I know that there are, other than the examples that I showed, you know, this is a tough one for me to answer. Yeah, we try to avoid moving huge data sets. When we do, we just use sort of like the normal protocols. But definitely, I'm sure that there are many people who are working on more effective and efficient ways to actually transfer data. One thing that we've done too to try to avoid transferring data, of course, is try to work on other technologies like remote visualization and so forth. That's sort of been our, that's been one of our major efforts.
So thank you. I think moving the computation and the visualization to the data is really what is more common now, or what will be even more common in the future.
Yeah.
So one more question. What do you think about the common workflow language, and does Kepler support CWL?
That's another very good question. I wish Ilkay was here to answer that. I'm pretty sure it does. I generally think common workflow language is going to be a good move for the community. I don't know when we have actually the hard stop here. I do believe it does support it, but I'm not sure exactly to what extent it does.
Okay, I do think we need to stop now, but thank you very much, Rommie. This was a wonderful talk, and we really appreciate you coming here and sharing your expertise with us.
So, you're welcome. It was fun. Thanks.
All right, goodbye. Thank you all. See you next week.